Is Nelson the only one here?
Just a little bit of lag. I'm checking.
No, but I think I remember.
Started it? I'm still searching. There still might be a lag.
Although we have to we went on with the development concentration thing that never.
Okay, that's one. Okay.
RNA virologist, he's pivoted quite effectively with SARS-CoV-2. But the other company is taking time.
I know, yeah.
Is faculty member of the department who works on prescriptional changes during facility division. There's also started out studying changes. Now he's really getting into so the project's been Oh, okay, great. Okay, so we can get started. I want to welcome everyone to this seminar, which is a joint invite between Nelson and myself, and so this is jointly sponsored by the Genome Science Institute. Today we are going to welcome Bess Frost, who is the Salome Thoreau Director of the Center for Alzheimer's Disease Research and Professor of Molecular Biology, Cell Biology, and Biochemistry and Brain Science at Brown University. I see this department as a combination of cell biology and biochemistry also. Bess did her undergraduate degree at the University of Texas at Austin, and then she did her PhD working with Mark Diamond, who was at that point at UCSF. Mark is well known in the field for his work on tauopathies and cell models for looking at the transmission of tau. And that's the work that Bess did in his lab concerned looking at tau strains and ways that tau can transfer from one cell to another. Bess then went on to a postdoctoral fellowship with Mel Feeney at Harvard, who is a well-known Drosophila person who studies neurodegeneration, and Bess did some really nice work looking at the changes in chromatin during neurodegenerative disease in fly models, and also impact of on the nuclear envelope. And then she became a faculty member at University of Texas Health Science Center in San Antonio, rose through the ranks to associate professor, and then just this year moved to Brown to assume her current physician. So Bess has really established herself in a very interesting area in the tauopathy Alzheimer field, which she'll tell you all about, looking at retrotransposons and the role of retrotransposition in the pathology of the disease. And this is really interesting. It's quite different from some other areas in the Alzheimer field. So I think it's going to be really interesting to hear from her.
Right. Thanks, David, and thanks to you and Nelson both for inviting me and hosting and organizing today's meeting. It was really hard thinking, I was asked for a list of faculty to meet within the department. I'm going through even people in the department who are not in Alzheimer's and related dementia. There's so much overlap between what we do on the effects of tau on chromatin and RNA biology and these endogenous retroviruses, for those of you who work in virology, that I was really excited to come talk with you today. All right. Okay, so in terms of where we are, So for Alzheimer's disease, we previously had to depend on a post-mortem diagnosis for diagnosing someone with Alzheimer's disease. So the defining characteristics were these amyloid plaques and neurofibrillary tau tangles. We're now at a point where someone can go in and get a brain scan, and you can actually see these plaques and tangles in brains of people while they're still alive. Just this year, there's major progress in terms of a blood test for Alzheimer's disease. It's not yet FDA approved, but we now have ways that really effectively can tell you how much plaque is in someone's brain. We also, in terms of therapeutic progress for Alzheimer's disease, there's been a big leap forward, although not great drugs. But the first Alzheimer's drug that actually do slow neurodegeneration have been FDA approved. So the original was aducanumab, then we had lecanemab, and now donanemab. So these drugs target the amyloid plaques, and they're very, very good at clearing out plaques. And there is some slowing of neurodegeneration in individuals who take these anti-amyloid medications. However, they can cause brain bleeding. You have to go in to the clinic depending on the drug, every 2 weeks or every month for IV infusion. So that really limits accessibility. They can really only be used earlier in the disease state, in the disease process. So you can't be too far along to get them. And then people who have the ApoE4 genotype are not recommended to go on these anti-amyloids because their risk for brain bleeding is higher. So exciting that we now have these drugs. It's the first time that we've been able to slow the process, but there's still a lot of room for clinical development in Alzheimer's disease. Okay, so in terms of why these drugs may not be working, well, we know that when someone comes to the clinic with cognitive impairment, they've actually been accumulating these plaques and tangles for decades before they're having any memory deficits. And if you take brain scans of people and you look in a longitudinal manner, and you look at where the plaques are and where the tangles are, and then you image those people 2 years later, you see that the plaques or that the tangles overlap really well with areas of the brain that are actually degenerating 2 years later, whereas the amyloid beta is kind of diffuse all over the brain. And this really correlates nicely with studies in the post-mortem human brain from Brock and Brock, showing that the plaques are kind of everywhere, but that the tau is really depositing where the brain is degenerating. So there's a lot of effort now in pharma to develop drugs that either prevent formation of the tau tangles, or once they're there, are clear it out, or just decrease overall tau levels with antisense oligos, for example. That's a major strategy. In terms of where I think scientists, what we're trying to think of in terms of drug development is, the amyloid, we can already clear it out very, very well. The tau is really being hit by pharma. What's going to be next in terms of drug development around Alzheimer's disease? And so thinking about that, this is really where my group is positioned. once you have amyloid and tau in the brain, what is that tau actually doing to cause neurodegeneration? So that's really been where we're focused. And in terms of focusing on amyloid versus tau, the tau deposits in Alzheimer's disease, but it also deposits in a bunch of other neurodegenerative disorders that are not Alzheimer's disease that don't have plaques. And we know that tau dysfunction is sufficient to cause neurodegeneration because mutations in the gene encoding tau cause these autosomal dominant frontotemporal dementias. So there's just two examples here, but there's a bunch of different tauopathies that are caused by tau mutation, and a bunch of other neurodegenerative disorders where tau is the primary pathological feature. These are sporadic, so there's no tau mutation, but the tau is accumulating in these disorders. So collectively, these are called the tauopathies, and among the tauopathies, Alzheimer's is the most common. But we think that by understanding what tau is actually doing to drive neurodegeneration, that these drugs that we would potentially develop could be relevant to Alzheimer's disease, but may also be relevant to the larger group of tauopathies that are not Alzheimer's disease. So before I started working in this area, it was already known that these pathogenic tau species cause an overstabilization of filamentous actin. So you normally have actin filaments, like if you're thinking about what I have diagrammed here. So you have the actin cytoskeleton filamentous on the outside of the nuclear envelope. And then here's the nuclear envelope. There's a complex called the link complex that spans the nuclear envelope that binds to actin filaments in the cytoskeleton, as well as these lamin intermediate filaments on the inside of the nuclear envelope. So the lamin proteins are, the lamin protein assembles into intermediate filaments that line the surface of the nucleus and give it strength and structure. They're also the lamin nucleoskeleton is also really important for serving as tethering sites for highly condensed heterochromatin. Okay, so when tau causes this overstabilization of actin, as a postdoctoral fellow, I found that the effects of tau on the actin through the link complex causes a destabilization of the lamin nucleoskeleton. So what this looks like, this is just pretty pictures, this is all published. What this looks like is in a neuronal nucleus from a control postmortem brain, compared to a neuronal nucleus from a brain affected by Alzheimer's disease. Instead of having this nice spherical nucleus, you get invaginations and blebbing of the nuclear envelope. And actually, I had originally found this as a postdoc in fly models of tauopathy and in human Alzheimer's disease brain, but since then, actually work coming out of BU from Ben's lab as well, has shown that these blebbing and nuclear envelope invaginations in tauopathy, they can happen in cells where you induce tau expression, they can happen in tau transgenic mice, they happen in iPSC models carrying tau mutations, they happen in human frontotemporal dementias that are caused by tau mutations. So it seems like this phenotype of tau causing these nuclear pleomorphisms is really well conserved across labs and across model systems of tauopathy. So then I and now others have also found that this destabilization of the lamin nucleoskeleton causes loss of tethering sites for heterochromatin. So the heterochromatin is no longer enriched at the nuclear periphery, and it starts to become more open. So again, what this looks like in a neuronal nucleus from postmortem human brain, the heterochromatin, which is in green, is really nicely structured within the nucleus. It surrounds the nucleolus, and then it's enriched at the nuclear periphery. But in the brain affected by Alzheimer's disease, not only do you see these like invaginations of the nuclear envelope, but there's an overall loss of heterochromatin and nuclear, like the three-dimensional architecture of the genome within the cell. So what I'll be telling you about today is why this decondensation of heterochromatin is actually driving neurotoxicity. So when you look along the chromosome at where this type of heterochromatin is enriched, this constitutive heterochromatin is highly enriched at the centromere and pericentromeric regions as well as the telomeres. And in general, there aren't as many genes in this type of chromatin, and the genes that are present in this type of chromatin are generally repressed. What is very abundant in this type of chromatin are these transposable elements. So we thought perhaps that because of the effects of tau on the actin cytoskeleton, which then have consequences on the overall genomic structuring, of DNA that it could drive the activation of these transposons. So when I first began learning about transposons, I learned about them in high school, and I thought that they were this like genetic oddity of May's, Barbara McClintock's work. And I was really shocked to learn how abundant they are in the human genome. So the human genome is only 2% exons. So when you do whole exome sequencing, it's really a very, very small fragment of the genome that you're looking at. About 25% introns, about 25% structural DNA, this would be like microsatellites. About 2% are the DNA transposons. So this is equal to the amount of DNA that's the exons. So for a DNA transposon, if it was active, it would be able to excise itself from the genome and then hop to a new location. So one activation event would cause a deletion and an insertion. So in humans, there are no, because of mutations that we have accumulated over the course of evolution, we don't have any DNA transposons that are still active. Almost half of the human genome are these retrotransposons. So for a retrotransposon, if it's activated, so normally or generally kept silent by heterochromatin and other factors, if it's activated, it's transcribed into RNA. Some of those retrotransposons are protein coding, so they make proteins that are similar to viral proteins, one of which can turn this RNA copy into a new DNA copy, which can then insert itself somewhere else in the genome. So similar to the DNA transposons, most retrotransposons can no longer do this entire process, but we do still have some retrotransposons in the human genome that are transpositionally active. Okay, so we first asked in our fly model of tauopathy if pathogenic tau changes RNA levels of transposons. So throughout the talk, I'll be showing you what we're actually focusing on here. So this is the RNA copies of the retrotransposons in the fly model of tauopathy. And we're looking here in the transcriptional profile in the adult fly brain. So you can see compared to control that there's a bunch of different retrotransposons that are elevated in the fly model of tauopathy. Most of these are of the long terminal repeat endogenous retrovirus class of transposable elements. So later we took 3 different mouse models of tauopathy and looked at, again, the transposable element transcript profile in these models. I'm just showing you one of the models here. So we analyzed the transcriptional profile at 3 months, 6 months, and 9 months of age in these mice. So by 9 months, they have neurodegeneration. And we see a really striking age-dependent activation of retrotransposons in this model, and this is also what we found in the other 2 models that we looked at. So anything going to the right here is elevated in the mouse model of tauopathy, and anything going to the left is depleted. And again, when we look at the class of retrotransposons that are getting activated, it's these long terminal repeat endogenous retrovirus class of transposable elements. So see here with the long terminal repeats, the ERV stands for endogenous retrovirus. Then looking at human brain affected by tauopathy, we had age-matched control, Alzheimer's, and PSP brain tissue. So PSP is one of these primary tauopathies that I mentioned earlier. There's no tau mutation that's causing it, but it's just pathogenic forms of wild-type tau in progressive supranuclear palsy. When we compare the transcriptional profile of transposons in these tauopathies compared to control, we see a bunch of different retrotransposons that are activated at the RNA level, a lot of overlap between the Alzheimer's versus the PSP, and again, it's these human endogenous retrovirus class of retrotransposons that seem to be activated. So we published our studies in fly models of tauopathy and Alzheimer's disease and PSP in 2018. At the same time, there was work from Josh Schulman's lab looking at a different fly model of tauopathy, as well as human Alzheimer's disease using a different publicly available RNA sequencing data set that found the same results as us. So we were nicely matched data there. And then in Josh's paper, they use the ROSMAP data set where you can look at transcriptional profiles and link them more to the tau tangle formation versus the plaque formation. And they saw that the transposon activation really aligns more with the tau tangle profile than the plaques. And then we found that these transposons were getting activated in tau transgenic mice, and then Tom LaRocca's group had complementary findings the same year. And then there was a group who was looking at transposable element transcript levels in blood taken from people in a longitudinal manner. So with these people, they were taking blood samples over time, and they were looking at what was changing around the time that people converted from normal memory to cognitive impairment. And they saw that there was a big spike in retrotransposon transcripts right before they converted to cognitive impairment. We've recently started working in iPSC-derived neurons and cerebral organoids, carrying this tau mutation that causes autosomal dominant frontotemporal dementia. So we can differentiate these into cortical organoids, iPSC-derived neurons. And when we look at the transcriptional profile, you can see a nice activation of retrotransposons in this iPSC-derived model. And we've also, using data from shared with us from Cat Bowles, we've also analyzed retrotransposons in iPSC-derived neurons carrying a different tau mutation, and we see that they're activated there as well. So taken together, it seems that this effect of tau on retrotransposons is really well conserved among model systems and labs. Okay, so that's looking at the transcripts. In our original work, we also looked at actual retrotransposition. So here we're using a fluorescent reporter of transposition where, when there is insertion into a particular hotspot in flies, we get green fluorescence. So this reporter was originally developed by Josh Dubnow. So in the control flies, we have this fluorescent reporter of transposition, the MEG4 trap. There's not very much green fluorescence there. But in the tau transgenics, we have a nice elevation of GFP, suggesting that there's more transposition in this model. So this analysis was done at day 10 of adulthood, when we have a good amount of neurodegeneration in the model. If we look earlier in the model, there's no difference between control and tau in terms of how much retrotransposition is happening. This is day 1 of adulthood. Day 10, nice increase of GFP, and then by day 40, it's sustained. So it seems to be an age-dependent activation of retrotransposition in this model. In unpublished work, we really want to know if transposons are mobilizing in the human brain, in somatic cells of the human brain. And we trialed several different approaches to see if this was happening. Each approach really had major caveats, and we got tired of those caveats. So we just went straight into long-read DNA sequencing. So in this experiment, we have post-mortem human brain of individuals with no Alzheimer's disease pathology, so that's called Braak stage 0, mid Alzheimer's disease pathology, that's Braak stage 3, and then late Alzheimer's disease pathology, that's Braak stage 5-6. We extracted DNA from those brains and then analyzed how much retrotransposition was happening in potentially somatic cells. So first, when we pool all the brains together to see the types of retrotransposons that are jumping and what they're jumping into, we see that the most abundant type of transposable element that's moving are these ALU-YA5 elements. These are like 300 base pair elements that rely on line 1 for their transposition. And then in terms of what they're jumping into, it's mostly centromeric DNA. When we compare this potentially somatic retrotransposition of ALU-Y elements, really restricting it to these signatures that line 1 mediated transposition imparts on the DNA, and we compare among stages of Alzheimer's disease, first of all, there's no significant elevation of transposition in the Alzheimer's cases. But you do see this trending elevation of ALU-YA5 retrotransposition. I have to say, with this analysis, I was generally underwhelmed by the amount of retrotransposition that's happening in the human brain. It's very clearly happening, but I think it's pretty infrequent, and other people have found this as well. And I think when it does happen, it's probably not, we don't have any evidence that it's really jumping preferentially into protein coding genes that are going to be really important for neuronal function. So it's happening. It may be increased in Alzheimer's disease, but I don't know how much it really matters for toxicity. So I'll get to what we're thinking about in terms of that in a few slides. All right, so can we drug retrotransposon activation? There was work in fly models of aging where transposons are also activated that use this HIV drug called 3TC. So 3TC is a nucleoside analog reverse transcriptase inhibitor. It blocks the conversion of RNA into a new DNA copy, and it was shown to reduce transposition that happens as a consequence of normal aging in flies. So we took the fly model of tauopathy, crossed with this reporter of transposition, and we fed them either vehicle or drug. And we found that the reverse transcriptase inhibitor caused significantly less transposition in the fly model of tauopathy. And then when we look at actual neurotoxicity in this model, while non-transgenic flies fed 3TC don't have any significant toxicity, the fly model of tauopathy has a lot of neurodegeneration compared to control, and then when we block transposition with 3TC, we have significantly less neurodegeneration. And we use a variety of other assays to measure neurotoxicity in this model, all of which were suppressed by this drug. So we published that in 2018 in Flies. In 2023 and 22, there were two different labs working in different models of tauopathy in mice that showed effective suppression of neurotoxicity using the same drug in tau transgenic mice. And then this year, work from Andrew Yu's lab using induced neurons of individuals with sporadic Alzheimer's disease. So they let these neurons form these neuronal assemblies. Found also that 3TC significantly reduces toxicity in that cell-based model of Alzheimer's disease. We have also tested 3TC in iPSC-derived cerebral organoids carrying this tau mutation. So I showed you previously that the transposons are activated nicely in this model at the RNA level. If we measure neurotoxicity in these organoids using bicleaved caspase, we see that 3TC nicely reduces tau toxicity in this model as well. Okay, so thinking about how frequently this is actually happening in humans and how much it's really going to contribute to neurodegeneration, we've kind of stepped back thinking about the overall life cycle of a retrotransposon and at the other products that a retrotransposon makes that are very similar to viruses when they're activated. So for example, retrotransposons can be bidirectionally transcribed, and those complementary RNA species can come together to make a double-stranded RNA, which would be recognized by the innate immune system as viral. They're also very repetitive. So if you have a bunch of elements in a line and they're transcribed into one transcript, that can fold back on itself and cause a double-stranded RNA as well. Another factor that we're interested in is these DNA species, if they're not inserted into the genome, which studies in flies suggest most of the time they are not inserted into the genome, they can exist outside of a genome and sometimes circularize. And this extra chromosomal circular DNA can also be recognized by the innate immune system as viral. And then the proteins that are present also look like viral proteins to the cell. So we're kind of thinking about all of these factors and their ability to drive an innate immune response. And I'll show you some of our recent work for double-stranded RNAs. So to initially think about these double-stranded RNAs and if they could be driving neurotoxicity, Elizabeth, a PhD student in my lab, took post-mortem human brain tissue from people lacking any Alzheimer's pathology, mid-stage, late stage, and then people who had progressive supranuclear palsy, and just did immunofluorescence to look at the double-stranded RNA, GFAP, which is a marker of reactive astrocytes. So this antibody recognizes double-stranded RNA stretches of longer than 40 nucleotides. So what she found was that not only was the double-stranded RNA elevated in the disease cases, it really, really nicely overlapped with these reactive astrocytes. We were not expecting that. And then this cells have several different proteins that recognize double-stranded RNA that can then go on to induce an innate immune response. Elizabeth looked at one of these, MDA5, and saw that along with the double-stranded RNA and reactive astrocytes, that the MDA5 was getting activated was elevated in astrocytes affected by tauopathy, suggesting that not only are these astrocytes, not only do they have a high double-stranded RNA burden, but they're upregulating machinery that detects that double-stranded RNA as well. Going into tau transgenic mice, she sees the same thing. Again, the double-stranded, so this is a mouse model of tauopathy. Again, the double-stranded RNAs are significantly elevated, and again, the high burden is particularly in astrocytes in this model. Flies don't have glia that's exactly like mammalian systems. However, they do have astrocyte-like glia. And Elizabeth analyzed total double-stranded RNA levels as well as double-stranded RNA levels in astrocyte-like glia, and saw that they were elevated in the fly model of tauopathy as well. So really suggesting that this tau-induced accumulation of double-stranded RNA was conserved all the way from flies to mice to humans. So we wanted to know if the double-stranded RNA that was accumulating was actually coming from retrotransposons. So Elizabeth made a nanostring code set that would detect RNA from transposable elements that we knew were activated at the transcript level in tau transgenic flies. And so anything going to the right and above the line is significantly elevated in the fly model of tauopathy. She took the antibody that recognizes double-stranded RNA, immunoprecipitated that RNA, and then put it on the nanostring code set. And so she saw 3 transposable elements that were significantly contributing to the retrotransposon double-stranded RNA burden. So since then, we've taken a more unbiased approach and immunoprecipitated that double-stranded RNA and sequenced it. So you can see here, everything in red are retrotransposons. Everything going to the right is elevated in the tau fly model. So there certainly are other factors that are other genes that are forming double-stranded RNAs, but you can see there's a general shift in terms of the retrotransposon. So they're certainly contributing to the double-stranded RNA profile in tauopathy, the fly model. Okay, so we wanted to know if this accumulation of double-stranded RNA was actually driving an immune response. So Elizabeth, again, made another nanostring code set consisting of transcripts that recognize a bunch of different mediators of neuroinflammation in flies. So this is the RNAi pathway, JAK-STAT, the IMD pathway, and the TOL was the last one, sorry. Okay, so when we look just comparing control to tau transgenic flies, so everything is normalized to control here, you see nice activation of a bunch of different immune regulators in the tau transgenic flies. She took the tau transgenic flies and overexpressed dicer2. So in flies, dicer2 binds to double-stranded RNA and then mediates its clearance. And saw that the tau flies with pan-neuronal overexpression of dicer2 have significantly less double-stranded RNA. So it was a good way to reduce double-stranded RNA in this model. And she looked at the effects of this dicer2 overexpression on these immune pathways. So here you can see she nicely overexpressed dicer2 and then got significant reduction of a bunch of different transcripts related to neuroinflammation in the fly model of tauopathy. And then when she asked if clearing this double-stranded RNA actually made a difference for neurotoxicity, she could get less tunnel positivity in the tau fly model when she overexpressed dicer2. So taken together, these data suggest that tau elevates double-stranded RNA, that clearing those double-stranded RNAs from neurons suppresses tau-induced neuroinflammation and tau-induced neurotoxicity. All right. We've recently started investigating these circular DNAs in the context of tauopathy. So we had found, we had published in tau transgenic mice that the DNA copy number of retrotransposons is elevated. So this, again, is a nanostring code set that Paul and Gabby had used in the lab that's detecting a bunch of different, it's able to detect a bunch of different types of retrotransposon DNA in mice. So we had originally created this code set because we wanted to know if this transposition was happening in mice. Of course, the code set is agnostic to whether it's detecting new insertions, the original DNA that the transposon came from, DNA that has been generated by reverse transcription and not inserted, as well as circular DNA. So we would be detecting all of that stuff with this approach. So when they took tau transgenic mice and looked at 2 months in their disease course, which is when these mice don't have any neurodegeneration yet, they only saw one type of retrotransposon that had elevated DNA copy number. So this is detects a few different line 1 elements. When they age those mice out to 12 months, you see a further increase in these line 1 elements, as well as significant elevation in DNA copy number of several different mouse endogenous retroviruses, suggesting that some of these species are elevated. So some of the elements that these retrotransposons encode are actually not retrotransposition competent, suggesting that some of the signal is likely not coming from new insertions. It's probably coming from DNA that is not inserted. So we've recently gone into postmortem human brain from Brock stage 0, so that's the control, Brock stage 3, 5, 6, and PSP, and isolated circular DNA and sequenced it. So you can see at the early stages of disease, there's a real shift towards increase in the disease case in terms of the burden of circular DNAs. Only one of them is hitting significance based on the adjusted p-value. We're only sequencing 6 brains of each genotype, so we're not really powered, but this is like clearly showing something's trending towards elevation. That's sustained in later stages of Alzheimer's disease, but we don't really see it for the PSP brains. So we were really excited to see this, and you can see here that these circular DNAs have a lot of transposons in them. So we were thinking, great, that the retrotransposons are reverse transcribed, it's circularizing, and that's what this is. But when we go into that data set, this is what we were expecting, that they are all retrotransposons, but what we actually see is that these circles are mostly, have like chunks of retrotransposon in them, but then the rest of the circle is just like bordering DNA. So our original model where these circles are, originating from a reverse transcribed DNA is probably not correct. And that while these circular DNAs are certainly elevated in the context of disease, it's probably not the mechanism that we were originally thinking about. So this is a kind of new project in the lab, but I'm pretty excited to see where it goes. All right, so I mentioned to you previously, like in terms of drug development, what can we do? And we already know that this 3TC, we know that it's safe, it's been prescribed since the 90s, we know that it suppresses toxicity in fly models of tauopathy, mouse models of tauopathy, organoid models of Alzheimer's disease. So in the early stages of the pandemic, I worked with Dr. Campbell Sullivan and Dr. Nick Moosey on a clinical trial, a phase 2A clinical trial in people with mild cognitive impairment due to suspected Alzheimer's disease. Sorry, that's out of order. Okay, so this was an open label 6-month phase 2A trial. We had 12 individuals who had cognitive assessments that suggestive of early stage Alzheimer's disease. And they took once daily 3TC at the normal dose that's given to people with Alzheimer's disease. And we were very surprised, because this is a small study that was not really powered for any type of efficacy, that we have significant reduction comparing people before a drug, and then after 6 months of exposure to drug, we have significant reduction of GFAP in cerebrospinal fluid. So GFAP, you may remember from earlier in the slide, is elevated in reactive astrocytes. So it appears that the drug, at least for this particular assay, is reducing neuroinflammation. We have trending reduction as well of neurofilament light in cerebrospinal fluid. So neurofilament light is a protein that's generally elevated in cerebrospinal fluid and blood of people who have different types of neurodegenerative disorders, so not just Alzheimer's disease, but general neurodegeneration. So the trending decrease in CSF is encouraging. However, we do have a trending increase of neurofilament light in blood from these participants, so we'll have to sort that out in a larger trial. And then a change that we were very surprised to see, because the mechanism that we're hitting is downstream of tau. We wouldn't expect for it to change tau levels or amyloid levels. We have a very slight but significant elevation of amyloid beta 42/40 ratio in plasma. So when individuals have a high plaque load in the brain, they have low A beta 42/40 in plasma. So the elevation of A beta 42/40 in plasma would suggest that they have lower levels of plaques in the brain. When we look at cognitive function in the individuals in our trial using the PAC-5 test as well as the Mini-Mental test, we have no significant difference in cognition over the 6-month trial. We would not expect to improve anyone in this short time frame. You usually need at least 18 months to actually see significant changes in cognition, and way more people than 12. But we were pleased to see that we were not making the group worse. You will notice that there are some individuals who have a lot of decline in cognitive function, and these were the people that had COVID during the trial. So you could really see that in the data. All right, so where we are, one of the ways that we failed for the trial was in terms of establishing target engagement. It's really difficult to establish target engagement for this type of drug. When it's been used for HIV, you can measure actual, HIV burden, reverse transcriptase in fluids. And so we originally thought that we could potentially measure reverse transcriptase activity in blood and CSF from individuals in the trial. So the way that this works is that, the way that the assay works is that within the tube, you have DNA, and then you're supplying it with whatever fluid you're analyzing. And if that fluid has reverse transcriptase activity in it, it will generate these DNA RNA hybrids that have been detected by a dye. Unfortunately, this dye also can detect double-stranded RNA that's present in that sample, which we didn't know was going to be elevated prior to designing this trial, and could detect any DNA RNA hybrids that were already in your sample before you added the potential reverse transcriptase. So the results for this were all over the place. You can see there's no significant decline in plasma reverse transcriptase activity. So we're kind of going back to basics here to try to figure out what we can measure in fluids that's elevated because of transposon activation that is responsive to 3TC. So Zach Utke is an MD PhD student in the lab who's kind of leading this effort. We originally thought that these drugs would they would be blocking this, they would be blocking this, and they would be blocking this because the reverse transcriptase inhibitor functions right here. However, because other people now have been working in this area, they have shown that these reverse transcriptase inhibitors also decrease retrotransposon RNA. I'm not totally sure how that's happening, but we're trying to figure out in iPSC-derived neurons carrying tau mutations, if we can look at things that are secreted into the media and see if 3TC changes double-stranded RNA, changes DNA RNA hybrids, changes episomal DNA, changes regular RNA transcripts, and changes transposable element-derived proteins. So we're really taking a holistic view to see what these drugs are hitting, if cells are secreting it, and then we'll go back to the fluids that we collected during the clinical trial to see if those are changing in response to 3TC. I think this is really, in terms of clinical development around this class of drugs, I think this is the real challenge. Okay, so we have finished this phase 2. That's the data that I just showed you. since we published our work on tau and retrotransposons and antiviral medications, others have initiated trials in the context of tauopathy using different HIV drugs. So this repurposing nucleoside reverse transcriptase inhibitors for AD, the LINE AD study is being led by John Sedeby and Megan Riddle at Brown. So same concept, also in Alzheimer's disease, but they're using a different HIV drug than what we used. And then transposon, I forgot to disclose that I'm on the scientific advisory board for Transposon Therapeutics. So Transposon Therapeutics just finished a phase 2A of a different reverse transcriptase inhibitor in people with progressive supranuclear palsy and had really encouraging results from their phase 2A. They granted them fast track approval from the, fast track designation from the FDA. So this is a primary tauopathy, hitting the same thing, different drug, different reverse transcriptase inhibitor. And then for ALS, in which transposable elements are also activated, another HIV drug that consists of a reverse transcriptase inhibitor, 2 reverse transcriptase inhibitors, and an integrase inhibitor, so TrueMech, had positive results from phase 2 and is now in phase 3 for ALS. So in terms of clinical development around this mechanism, there's a lot of activity for neurodegenerative disorders. All right, so that is where I will stop. I'd really like to thank the people who donated their brains. It's a really critical aspect of what we do in the lab, as well as the caregivers and participants in our clinical trial. The clinical trial was funded by the Owens Family Foundation. We actually did it for $70,000. So we're planning for the next phase of a trial when we would really like to be operating with a larger budget so that we can do all of the things that we want to do, have a placebo control and more participants. And I thanked everyone in my lab going through the talk, so I'll answer questions.
That was great. The circular DNA species you're sequencing, how big are those circles? And you mentioned that you don't see full-length transposons, you only see segments. Do those segments give you a clue how those circles are being formed? So how big are they?
Yeah. So the first question is, how large are the circles? And we have not quantified the size of the circles. We've just isolated circles and then sequenced them, but we're not doing long-read sequencing. It's just 150 base pair paired end sequencing. So we can't say the size of the full circle. But that's a very good question that I would like to know. And then in terms of the fragment of transposable element that's inside of them, we do see full-size alu elements within the circles for sure. So it's not always fragmented. Yeah. We did not, like for a line 1HS transposable element, for example, there was no full-length ones in there. So those are fragmented, but the signs are full-length. Interestingly, a lot of the circular DNA is coming from ribosomal DNA. So that was totally unexpected but cool. And there's work in yeast, actually David Sinclair's from like the late 90s, showed that this elevated circular ribosomal DNA drives aging in yeast. And there was some other work in that area for a while, but not a lot. There's really not much. I don't think they've been shown in mammalian. I could be wrong, but I don't think they've been shown in mammalian systems. So we weren't looking for it, but it was really cool to see this elevation of the ribosomal DNA circles in the AD cases. Yeah.
Ben?
So really presenting a bunch of questions, but I'll just say 2 first. So the transposons are related. Josh Duke now showed this in flies, so there's a clinical trial of ALS and AD. So it seems like it's kind of a general mechanism, So how do you fit that in with the idea of does selective therapy matter? Are these diseases like all the same thing, or is there this is general? Like how's that?
Yeah, so Ben's question is about this activation of retrotransposons across neurodegenerative disorders. Yeah, so they're activated in a bunch of different neurodegenerative disorders. They're also activated in the context of human aging. They're also activated in cancer. So I think one of the contributors to this activation across age-related diseases is this age-related decondensation of heterochromatin, which is important for activating them. However, I don't know if that's true for all of the disorders, because TDP43, for example, work from Josh Dubnow and Molly Hamill, has shown that the reason why retrotransposons are activated in the context of TDP43 pathology is because normally TDP43 binds these RNA transcripts and keeps them inert, I guess. So then when you have loss of TDP43 function, they can go on to do the rest of the activation. So, but I also wonder if tau might be doing that as well. Might be binding directly to transposable element RNAs. I've been wondering if you guys were thinking about that and doing that too. Yeah. But what's
the other thing just for is you put in those mechanisms of cellular response, right? So you didn't have CGAS in there.
Oh yeah.
CGAS is like the classic double-stranded RNA to interferon response.
Right. Okay. So what we're working on now is the double-stranded RNA sensors. So the double-stranded RNA is like PKR, MDA5, TLR3, I think. So we're trying to see if those factors are detecting and then causing innate immune activation. CGAS-STING actually recognizes the extra chromosomal DNA. So yeah, we're working on that as well. So CGAS is clearly activated in tauopathy. There was work from Lee Gann and I think now others in tau transgenic mice. CGAS-STING pathway is activated, and they propose that this is because of leakage of the mitochondrial genome, which they clearly show that mitochondrial DNA is leaking into the cytosol in the tau models, but they don't actually link the mitochondrial DNA to the CGAS-STING activation. So it's possible that the retrotransposons are contributing as well, and so we're working on this a lot now. Yeah.
Nadia.
I was really curious actually. Did you find any retrotransposons to maybe 3R or 4R specific, or have you found that people with different tau strains to begin with may have different retrotransposon activation?
Great question. So the question is about tau that harbors 3 microtubule binding regions versus 4 microtubule binding regions. So there are certain tauopathies that are composed of 3 repeat and 4 repeat tau fibrils. Some tauopathies are all 4 repeat tau fibrils, and some are all 3 repeat tau models. So all of the model systems that we're working with, the fly and the mouse model systems that we're working with in the lab are all 4 repeat tau. The iPSC model that we're using one that pushes tau more towards 4 repeats, although there is some 3 repeat there as well. And then the human Alzheimer's disease cases, those are 3R, 4R, and then PSP is a 4R. So the retrotransposons seem to be activated in all of those models, but we've never looked at a model that's only 3R, so I can't say that. So for example, we could look at Pick's disease to see if transposons are activated there, but yeah, we haven't, and no one else has to my knowledge.
Back there.
2 questions. The first one is about the double-stranded RNA. You nicely showed double-stranded RNA covering mutation and also MDA5. Did you ever see MDA5 pulling down these retrotransposons double-stranded RNA?
Yeah, we haven't done that, but we plan to. It's in my R01. I hope you find it.
Just to follow up on the circular DNA.
Yeah.
The DNA ends are what are needed for immunostimulatory activation. If they're circularized, I'm puzzled as to how you think circular DNA can induce immune activation.
I believe CGAS can bind to double-stranded DNA. To circular DNA. Is that right? Okay. Well, we need to rethink that in that case. But it seems that the circular DNAs aren't mostly transposable element anyway. So yeah. Julia.
Yes. If you think about developmental biology, these neurons, this is normal, like canonical mechanism that neuronal development, like transposable,
Yeah.
And the neurons have deep learning and memory as well.
Mm-hmm.
And do you think, it's completely hypothetical, but do you think this thing happening related to disease is due to the fact that other glial cells cannot handle properly to clear up for misfolding one, or is this you compare like the one that actually reported in a book about like neuronal development versus this disease specificity of all these.
Yeah, so the question is about So in neural development, in developing neurons, they also activate transposable elements. They also have more open chromatin in developmental states, and they can divide. So I didn't mention this previously, but this pathway that I've been talking about also leads to abortive cell cycle reactivation in neurons affected by tau. So tau causes a lot of these developmental phenotypes, except for that the postmitotic neurons die instead of actually divide. So I think, I have a review article about this actually, I think that tau is causing neurons to lose their terminally differentiated states. And they're basically de-differentiating to a more immature state, but something about mature neurons causes them to die rather than to divide, probably to protect the brain against cancer. You also asked about how non-neuronal cells can contribute to this, and I don't know about that part.
Oh.
Yeah, to ask also about the actual retrotransposition events. So can you register them somehow just to see that, okay, we see some insertions of the transposons in the new regions of the genome that can cause or maybe even like stimulate faster development of the Alzheimer's, except that the mechanism, biochemical mechanism.
Yeah, I mean, we looked into that, but most of them are jumping into centromeric regions. So it's just like repetitive DNA, and there's no program to tell you how important that is. So we don't, I really don't think it's driving toxicity. Yeah. We see a lot of problems at the centromere and pericentromeric region. So we see, based on the long-read sequencing, in the disease cases, there's less DNA methylation, there's more recombination events. So there's definitely bad stuff happening in the centromere, but I don't think it's because retrotransposons are just jumping into it so much. I think we would detect more than what we detect, but I could be wrong.
Mike.
So work from a couple of labs have shown that stress conditions can induce mitochondrial double-strand RNA. So I was wondering if you've looked for bidirectional mitochondrial transcripts in any of your tau models?
We have not, but we have the data sets where we could go look at that specifically. I did not know about that. That's a good idea, and we can definitely check.
Okay, Valentina.
Attaching this, have you looked whether there is any increase in new mites, mitochondrial DNA inserted into the genome? Because they also jump into the nuclear genome, and particularly in centromeric areas. And it's something that is happening in the brain, not elsewhere. So those are events that before were thought to happen very rarely, but now people have reported them.
I was not aware of that, and we didn't look at it, but we can go back and check. Yeah, that's
That
cool.
would be something.
Yeah.
It's interesting that you have questions from, as you say, from people with a variety of expertises that are related to what you're talking about.
Yeah.
So, okay, no more questions.
Thank
That was great.
Pack all this up. Did you bring the bag? Yeah, okay. Pack everything up, bring it back to the room. Yeah, stream worked well. There are 5 people. Nice.
One of them is shooting.
Okay.
He texted me and said everything.